So we're only going to go through maybe the first couple of items: enzymes, regulation of enzymatic activity, maybe a little bit of energy, but we won't get too far into it. And so wherever we stop, I can give you the page probably when we're done. I mean, you'll see what we do is what I'm allowed to cover on Monday. But this is much bigger than just that test. But this is really important. And like I said, the problem is some of you probably get bored with it. A minority is going to get bored with this because they've heard it before or multiple times before. For some it's new, so I have to find a way where it's in the middle, where nobody gets too bored or other people get too overwhelmed because so many things are taken for granted. So it'll get into basically metabolism means you're somehow trying to use nutrients to drive all the energy using processes in the cell, and what the cell does is it tries to maintain homeostasis, and so a lot of the processes are needed or need energy, and that's what all of this is about. And there's different ways to acquire energy and to produce energy. So enzymes play a huge role in. We always talked about or we talked about it a few times now about proteins and their prevalence in the cell. So that's where a lot of the proteins are enzymes that make these processes possible. So metabolism really includes all the chemical and physical workings of a cell, whatever the cell is doing. So you can imagine that never shuts down. The metabolism never sleeps. And you have two types of chemical reactions. That makes sense too. It's just like photosynthesis is building these sugar molecules, and then the exact reverse happens in consumers that use the sugar and make and use that for energy and go back to CO2 and water. The same kind of thing happens. You can imagine that there's a degradation, so there are molecules coming into the cell, and they have to be degraded, or they have to be broken down. So that would be catabolism or catabolism, and then you have anabolism, which means it has to also be, which is the opposite. It has to be, new structures have to be built. So if you think of the basic, you take a glucose, or you take an even smaller molecule, and or take the parts of take the single amino acids, and they're part of proteins. They have to, if a big protein is there, it has to at some point be taken apart, so you have single amino acids. And then some other component needs a big protein, so it has to build amino acids to protein. So you always have this continuous circle of building and destroying, or not destroying, but building and using up. Taking apart, building, taking apart, building. So that's those two processes. So the catabolism is degradative, breaks the bonds of large molecules, and it releases energy. So the energy is contained in these. There's a lot of different energies out there, but really in the cell, what the energy that the cell uses is the chemical energy, or the energy that is in the bond of molecules. So to remember that too. It's important. There are other possibilities, like in photosynthesis, but basically most of the energy comes out of breaking the bonds of larger molecules. That's where the energy is, and then so that releases energy. That releases energy that's in those molecules. But if you want to build a molecule, so anabolism, if you synthesize, you obviously need energy to go in, because there's no bond to break, there's no bond to break to release energy. So you would look at this here and say you have these bigger carbohydrates, lipids, proteins, and to make them into end products like CO2 or water, small molecules, end products, that releases energy. And then that energy is not really stored or anything, the energy has to be used. So the cell is using this energy that it uses from taking these apart immediately to build something else. There's a constant, it's not that it's stored somewhere, it's just there's molecules like ATP or NADH that are using this energy that they acquire from these breakdown processes to immediately build. So you have you have amino acids and you build a protein, you have simple building blocks. And that makes sense, right? It's what goes on every second of your life in your own body. So enzymes are really crucial to this whole process, and what they are, they're biological catalysts. So they're increasing the rate of the reaction, they're lowering the energy of activation, so that those reactions usually, those reactions of building or these reactions of breaking bonds or they may all happen over a long period of time, but enzymes help activate those, induce those reactions. So they're really, really crucial for this process. And they're actually they're not altered in the reaction. So they're not really they're part of the reaction because they facilitate that reaction, but they're not really part of the actual reaction in terms of they're supplying structural support. They're as it says up here, they lower the energy of activation, but they're not part of the actual reaction. Their atoms or elements or molecules are not going into the product. So they're providing a physical site for specific substrate molecules, and what it really does is, if you have an initial state of a reactant, so this would be the energy level. So there's a certain energy level, and the product of those, say you have two substances react, bonding to each other. That could happen because the state of that end product is lower, energy-wise. This could happen, but it really doesn't happen at the right pace and at the right time without enzymes. So this whole differential between here and here is the energy of activation in the absence of enzymes. So the enzyme just lowers this energy, it just simplifies the reaction. It makes it facilitates the reaction and makes it faster. In general, I mean, there's calculations of how much they increase the speed, and it's amazing. So that's just what you got to remember. They're proteins, and they're not really part of the product, and they're not altered, but they lower the activation, the energy of activation in a reaction. And a simple enzyme would consist of a protein alone, so that'd be a simple, but a lot of enzymes contain protein and non-protein molecules, and that's where those cofactors come in. So a lot of times you have what they call apoenzyme, a protein portion, so that's a conjugated enzyme. So like that I would say is the enzyme you should think of. That contains protein, which is the apoenzyme, and it has a cofactor, and that's a non-protein portion. And a lot of times those cofactors are iron, copper, magnesium, can also be organic molecules, vitamins. So those are basically just factors needed in addition to this big, usually much bigger protein than the substrate. So whenever a reaction happens, you have a reaction of specific chemicals or substrates, and the enzyme is much bigger, but it has a site where all this happens, and it's usually where the cofactors are located. And those cofactors help, and I show you in the next slide or in the next few, help with the reaction. So you have different ones, as I said. It could be metallic, and it could be organic. So that's where vitamins come in. You have to take in vitamins because they're part of a lot of enzymes, and that's important because everything is done by enzymes. So there's a few examples. Just, iron is important in our diet. Guys, we always had commercials. Nutella was always, that gives me the iron. But you guys didn't eat Nutella growing up, right? Does anybody eat Nutella now?
Yeah.
Very good. We grew up with Nutella, and we wanted peanut butter, and there was no peanut butter anywhere. Like, peanut butter is impossible to get because nobody grows peanuts in Europe. So you would, what you paid for, maybe not anymore, but what you pay for, used to pay for Nutella, a ridiculous amount of money, was what we paid for peanut butter. Like the smallest thing, we got it from Ireland. It was like $6, and it was like this big. Until my mom found she found an army base in our town, and then just stood around there now for them to bring out there was a PX, I think they call them, or something. She brought home one of those, which I still send them home now for Christmas. Like, here's some peanut butter. Yeah, like, it's pretty cheap. It's a pretty cheap Christmas. It's pretty cheap Christmas present. That's the way it should be. True. So iron is big, and supposedly that was always big. Nutella was full of iron. That's what they told us.
Sugar.
I know, but that's what we were told. So they were looking out for our enzymes. That could be a bonus question. Something about Nutella and peanut butter. Copper, magnesium, nickel. So a lot of those, zinc. A lot of times you're trying to get some of that, and so it's all for various important enzymes that have certain actions in your cell. And so those are the micronutrients, the nutrients that you don't need a whole lot of, but they're crucial for your nutrition. Vitamins are part of it too. So you have specificity, just like, for example, proteins and the cell membrane only bind to specific, or only allow the binding of specific molecules, and then they go into the cell. You have enzymes with a site that's specific for a certain substrate. So, and that makes sense if you think of the structure, the sequence of amino acids was the primary structure of a protein. Remember, then there was a secondary, tertiary, and quaternary structure, which meant the way they were basically had bonds, a lot of times sulfur bonds or hydrogen bonding to give that strand a certain shape in space, and then if you had multiple strands actually together, you would have the most complex proteins. And in these complex shapes are sites where the substrates come together. So they basically these proteins create meeting points for whatever reactions or reactants or substrates need to react. That's what they do, and they can be very complex. They can be a little bit more simple, but that's where the active site sits, or the catalytic site. So the area where those reactions take place. So there usually is so say this is the substrate, and in this case it produces two products. So in this case it's not forming a product, it's taking a substrate apart. In this specific example, so you would have a big enzyme, and it's specific to the substrate because another molecule that tries to dock on or use the active site won't fit. So the substrate is the enzyme has an active site that's specific for this substrate, and it creates a temporary complex called the enzyme substrate complex, and then it releases the product. And sometimes one of the theories is that enzymes are actually helping by adjusting to the substrate. So there's a substrate, I guess, induced formation of the active site. So the active site is usually specific to a certain substrate or molecule, and it actually can even change when this happens to the shape of this substrate. But all you need to remember really is that they have active sites and they're specific to a certain substrate. The induced fit would be when the substrate moves in and the enzyme actually basically tightens the active site around that substrate. So the cofactors, the irons and other metals, for example, or vitamins, are really important. There are those micronutrients we talked about, and they act as carriers a lot of times to assist the enzyme. So you have basically a big enzyme of protein, apoprotein, and you have or apoprotein, and you have a carrier that is attached to it. So it's just you can imagine it like a small assistant, and that's the cofactor. And what it does is, when, for example, here you have a substrate 2 and a substrate 1, so you have basically in some reactions, the reactions are all different. So in this case, you have two substrates where a certain chemical group is transferred from one to the other. That could be any metabolic or any kind of reaction that happens in metabolism, where the pink one has a chemical group that has to be transferred. There's a lot of transfer of groups to substrate number 2, which is green. And so that by binding into the enzyme and then attaching to that blue coenzyme, it enables the transfer of the chemical group from pink to green, or from substrate 1 to substrate 2.
Transferring chemicals?
Yeah, it's a chemical group. It could be in this case, I think it's CH. I don't know if that's what that stands for, but you could imagine it could be like, say, any kind of. I think we're going to go into examples, but any kind of chemical group, not necessarily single elements, but you have an amino group on a protein or on a amino acid that can be transferred, a carboxyl group, things like that. It's just depending on what it is. It's just an example to show you that in general you don't just have a protein with an active site, but you have cofactors, which are vitamins or metals, that facilitate that actual transfer from a group from one to the other. And interestingly enough, you have the action can happen in the cell. So that is happening, or what you would call then the enzymes are endoenzymes, and they retain the intracellular function. So most of them are happening in the cell. You would imagine that all the metabolism that goes on should happen in the cell, but you also have exoenzymes because they're transported out of the cell, and they for example break down large food molecules or chemicals or other compounds, and then they can enter the cell. And there was yesterday, I remember the day before yesterday, we had a picture of that where the food particles were too big to cross the membrane, and so the organism would release enzymes into the environment. And I think there are, what are there? There are some species, aren't there spiders, bigger spiders that actually, maybe it was tarantulas, but I thought they're actually releasing enzymes into with their saliva into the prey, and then they kind of start digestion of certain groups.
Flies do that.
Who does?
Flies. Like they regurgitate on the
Could be. But there it's happening, and so this would be a similar cellular process where enzymes are released into the environment, and then the products can come in because they're broken down. So they can easily make it through the cell membrane, or be transported into the cell through the cell membrane. And what's important, obviously, is how those enzymes are regulated, because you would say, well, say you just had a meal, and your glucose levels, your levels in your blood are going up. There's changes all the time, like how fast should that be stored, how fast should that be digested. If you only have a certain amount of enzymes, how are they going to keep up with this? Are you going to have a huge spike in blood sugar because they can't take it out, et cetera, et cetera? And that's just one example. So they have to be regulated, and there's two different kinds, constitutive enzymes and regulated enzymes. And those are always present. That means they're always produced at about the same rate. There's always about the same amount of those. And it doesn't really matter how much substrate is out there. And that's for very common processes where you know that, I think in glycolysis you have some of those. So you're always going to have to do a certain processes related to using up glucose, etc. So those would be always around and always at about the same rate. And then even if you add more substrate, it really doesn't change anything. That's what this is supposed to show you. You have a certain amount of enzymes in here, and you have the red substrate. Those are connected to those substrates in this case, but there's not an increase visible. And in regulated enzymes, it can be induced by certain signals. It could be the food particle. It could be a certain signal that induces the enzyme or induces enzyme production or turning off enzyme production. So in this case, you have more substrate, and then you have more enzyme. The enzyme levels increase with the substrate, and then the opposite would be you remove substrate, and the enzymes go, or the amount of enzyme decreases. So they're regulated. It's a pretty simple concept. And then what I wanted to, maybe should have brought that up a little bit earlier, but what's important for those anabolic and catabolic reactions is, so if you build or if you break down, it's connected with water, and so you call it, when it's synthesized, the condensation reaction, and when it's broken down, it's a hydrolysis reaction. And that's just another way of looking at it. But if you remember, a lot of times, here it's the glycosidic bond or glycolysis. So here you have glucose, you have a molecule of glucose and a molecule of glucose, and the enzyme facilitates the reaction. So it's starting to build a glucose chain, which could become a starch, or it could become a cellulose molecule. So if it takes these simple compounds in general, by creating this bond, takes energy. So we said when we build something, we need energy, and that energy is provided by ATP. And by creating these bonds, water is given off. So that would be a condensation reaction. So they synthesize, they have an anabolic reaction. So that kind of is synthesis, anabolic, and that usually creates a molecule of water that you see here. And it needs energy. So there are a few things in it that you should know. If you want to synthesize, you usually get you usually need energy. You build, so you need energy, and that's supplied by ATP. Water is released, and it's an anabolic reaction. And then on the other hand, just like we said before, on the opposite, you have a catabolic reaction, meaning you're not building, you're breaking down, and you need to put a water molecule in to make that happen, to separate those two amino acids, in this case, from each other. In this enzymatic reaction, you would have to put there's a need for a water molecule, and so these catabolic reactions are also hydrolysis reactions. Water is needed. It requires the input of water to break the bonds. And in this case, you generally are able to produce energy. So, for this question, an enzyme, does it become a part of the final product? No, it is non-specific for a substrate. It's usually specific, so it only does a certain... Is it consumed by the reaction? No. Is it composed only of protein? Don't forget the Nutella. No. So it has to be D. So you didn't even have to know that it can be active outside the cell. You can just exclude all the rest. That's how simple the multiple choice questions are. Let's see, how much do we have? I don't even know what stage. Oh yeah, we're gonna be able to almost make it to where I want it to be. So one of the things you've seen before is, you change the temperature in the growth of microorganisms, and they don't grow as well anymore. Why does that happen? And it a lot of times has to do with enzyme activity. So when you heat things up, or when you cool them down, enzymes don't function as well at those temperatures, or there's denaturation happening. So the environment of the cell is really important for the activity of the enzyme. So certain temperatures, pHs, osmotic pressure, all play into how well the enzyme works. So when there's changes in the habitat, usually that changes the enzymatic activity, and it could lead to chemically unstable enzymes or denaturation. Weak bonds that maintain the shape of the apoenzyme are broken. So whenever you hear denaturated proteins, you apply heat, and basically the enzymes, the shape of them, if you think of like a structure, how they're in space, and that changes because of the bonds breaking, etc., etc. You have proteins that are shaped weird, and they're not going to be able to, they're not going to be able to function. And if that happens enough, you've got all this protein garbage in a cell, and it dies. Or you have a lot of protein that can't fulfill its role, enzymes that can't fulfill their roles, and the cell dies. So denaturation is important, especially if we want to get rid of certain processes, say microbes, etc. So we have to figure out how to do that. And denaturation can happen through pH changes, temperature changes, osmotic pressure changes, things like that. So this is just to show you what usually happens, and we'll have some examples in metabolism, but you can either have, a linear system where you start off with glucose and there's a reaction, you have a next product, and an enzyme facilitates the next reaction, the next. So you have a cascade of enzymes working to make, to guide substrates through a whole process. So it's never just one substrate, enzymatic reaction, final product. So usually it's a cascade or a linear line of enzymatic reaction. It could be cyclic, which means you would have a certain product, or you have a certain substrate, and that goes into an enzymatic cycle, and then it becomes at some point you have a product, but then it can cycle back to its original form, usually through energy input, and then it cycles through that again. And there's some important cycles in metabolism. And then it could be branch, either divergent or convergent, depending on either you start off and you have two different pathways, or you start with two different pathways and they converge. But that's just a basic pathways, and we're going to go through different ones going through the metabolism. And like I said, if you wanna if the cell needs to regulate enzymatic activity, one possibility is you induce you like you've seen before, you induce the production of enzymes, so you just make more of it. But you can also inhibit the enzyme itself, and a lot of times, you have competitive inhibition and non-competitive inhibition. So that happens on the enzyme itself. So you have an enzyme and it's inhibited, which means it's a faster process. So if your enzyme activity is important, or it's important to change the enzyme activity quickly, you can. There's processes called competitive inhibition and non-competitive inhibition that make that possible. In the long run, you would probably want to, have induction where the gene expression, the protein biosynthesis produces more or less. But this is more of a fast solution. And what happens is, in the first case, you just have a substrate that resembles the normal structure and it competes at the active site. So here, if you think of acetylcholine that I showed you yesterday, there's other basically what could happen is you have a substance that comes in, it can actually bind to the sites that acetylcholine wants to bind at, and it can't bind anymore. So it's competing for that spot, and that can create big problems. It's depending on where that happens. But just remember, there's competitive and non-competitive. Competitive means this competitive inhibitor has a similar shape and can block, so no more space or no more active site for the actual substrate, so that's blocked, and the reaction can proceed. And then in non-competitive, there's a regulatory site. How we were taught is, there's some other molecule coming, and it basically attaches to the protein and it changes its shape. And then it can't, so it changes the shape. You can imagine, like, it bumps in there, the protein folds, and the active site is occupied or can't be reached anymore. But what it really does is you have a regulatory site and you have a regulatory molecule. So basically, when anything binds here, it changes it so that you see the shape here. That active site has changed its shape. And a lot of times what happens is, this is a product. So what happens is, if you imagine you have a certain substrate coming in, and normally it just binds. And then after binding, there's a product. And depending on how much product there is, some of those product actually binds on the next enzyme here and makes, basically prevents more product to or more substrate to be produced, and what you would call it is like a negative feedback. So it means if you have a lot of substrate being produced or a lot of product being produced, some of that enzymatic action will be decreased by that product being able to inhibit it. So negative feedback usually means it controls the reaction, so it doesn't get out of hand. There's some, and I'll have it up here. So the enzymes are regulated by the binding of a molecule, sometimes the product. It's got to be other than the substrate, away from the active site. If it's at the active site, it's competitive. If it's away from there, it's non-competitive. It doesn't compete for the active site. So enzyme repression would be on a genetic level, so how much is being produced, and induction would mean they're only made when a suitable substrate is present. But just remember that that's sometimes important in negative feedback. And I think we should. This is just an example. I'll stop with that. So just so you know, here, for example, you have a DNA strand and RNA is being produced, protein is being produced, and then you have an enzyme, and then this product of the enzyme blocks the DNA, so it can't produce more of that enzyme. And so that would be a perfect example of a negative feedback, where the more product you have, the less it's going to be produced, the enzyme, and then when you have less product, this all falls apart and it can produce enzyme again. You can look up negative feedback because it's an important and interesting process. That's it. Thanks for